Morning, Mackenzie. How are you?
Morning, Prita. How are you?
Good, good. Nice to see you again and connect.
Yeah, you too. How have you been?
Good, yeah. It's a busy time of year, but yeah, everything's good though.
Yeah, good.
Hi, Jade. Nice to see you too.
Hey,
Yeah, how are you?
good. How are you doing?
Good, good. So
Cool.
you're in Sarasota, is that right?
Yeah, I am. Yep.
Nice. That's cool. I'm sure the weather's a lot more desirable there than up here.
Certainly in the winter. Yeah,
Yeah.
like the ideal time. But in the
Yeah.
summer, it's
It's
hell.
miserable too. Yeah, it's like a
Yeah.
cold. Winter isn't like a cold place. So. You can't go
Yeah.
outside. Yeah. Yeah, that's true. Yeah, I had messaged before. I used to live in Fort Myers, so it was like close to Sarasota.
That's so cool. Yeah, my dad grew up
So
there.
random. That's cool that y'all are able to still work together. That's fun. I'm
Yeah.
excited to hear your developments and stuff. So
Cool.
yeah, but nice. Yeah, I just made both of you co-hosts.
Okay.
And then let me just pull up your bios. Yeah, we'll just get started right away. And then I'll introduce you and then I'll just pass it on to you. And then same as before, you can just do your presentation and then have questions and stuff. And then, yeah, we can aim to wrap up, I think, by around like 10:15. So like including with questions and stuff, something like that.
Okay, great.
Yeah.
So we'll try to be done by like 10 for 15 minutes of questions.
Yeah. It sounds
Okay,
good. And then let me just
great.
look. Mackenzie sent me your updated bios. Let me pull that up. And
Yeah,
then,
I don't know how old the one you have
yeah.
is, and I feel like a lot's
Yeah.
changed in the last couple of years for me, so
That was like at least
yeah.
2 years. So yeah, for sure. Um, and then Jade, is it okay if I just use the same one from you before?
Yeah, probably.
Okay. You can
I
feel
just,
free to, yeah, say
yeah, I
anything.
just live in Florida now. That's pretty much the big
The
change.
big change, yeah. Did you grow up there?
I did,
Yeah,
yeah. Yeah,
nice.
I moved
So,
away like 10 years ago.
hmm So want to be closer to family?
Yeah,
hmm Yeah.
yeah.
Nice. Cool. Okay, I'm just opening it up now. All right.
Just a heads up, I just got a little notification that my internet connection is unstable, so um
Okay.
we'll
Yeah.
see, but it got glitched out for a second there. Hopefully
Okay.
it holds up for us, but yeah.
Okay, no problem. Yeah, if you need to like turn off your video, then sometimes that helps.
I
Feel free.
might do that at some
Yes.
point.
Yeah.
Yeah.
Okay. Right. Cool. All right. So yeah, if y'all are ready, I can start letting people in, and then we'll just kind of get started. Okay.
Yeah.
Mm.
Jade, you were sharing, right?
Oh, I thought you were because of my.
Oh, yeah. You're right.
But there is that one part where, yeah, it'd be cool to click around the website if possible.
Letting folks in. So welcome everybody. Yeah, see we have. Oh yeah, we have some guests too. Um, yeah, welcome everyone. We're gonna just pretty much dive right in because the students that are in my class, we have a very full class today. So I want to like make sure we kind of get the most out of every moment. And then welcome to all our guests today too, to our special presentation from the folks at Metamimicry. So welcome everyone. We have mostly people from the Fungi program, but then there's a few folks from Evergreen staff and faculty and other students that are here to see your presentation too. So thank you, Jade and Mack. So yeah, I'm just going to introduce our guest speakers and then pass it on to them and we'll kind of dive right in. So first we have Jade Swor. Jade is the executive director of Metamimicry, whose mission is to explore accessible biological solutions for our time's most pressing environmental pollutants. And their vision is to shift towards a culture of remediation for diverse natural ecosystems and for humankind to thrive. One of their projects is also called the Spore Project, which works with the Squaxin Island tribe to support the tribe in growing mushrooms, and they'll share more about that work as well as their other work around bioremediation as well. And Jade, who uses any pronouns, is from the Gulf side of Florida, and has an ever-growing interest in the world of fungi. From studying applied cultural anthropology and sustainability at Evergreen, Jade then received a master's in environmental management from Western Colorado University in 2020, while starting a mushroom cultivation operation with the Potter Valley Tribe. And Jade's current passions lie at the interface of tribal relations, mycology, and biomimicry. And so welcome, Jade. And then also Jade's partner, Mack, is also another Evergreen graduate, graduated in 2019 with an emphasis in environmental chemistry and science communication. While working at a local mushroom farm, she was inspired to start a bioremediation group after a discovery of a new toxin, shortly after she met Jade and formed Metamimicry in 2021. Oh, and I'm sorry. Yeah, I should be reading. Yeah, I think I am reading your updated one, right? I just got confused for a second. Okay, am I good? Okay.
You're doing great.
Okay. Just a few months ago, Matt completed her Master's of Environmental Studies degree at Evergreen, where she pivoted into environmental policy. Matt currently works for the Department of Ecology in the Water Quality Department, working on the Puget Sound Nutrient Reduction Grant Program. She's driven to ask big picture questions and enjoys dynamic problem solving. So first let's give our guests a warm welcome, and my students too, if you're able to turn on your camera and say hi also. I know that usually helps our guests feel more welcome as well. And then, yeah, I understand if you need to go off camera for a moment, but thank you. Thanks, Moshe, Jess, and also my Gregor, Melissa. Nice to see you, Angelica, Benjamin. You, Nico, nice to see you too. But yeah, that means both of you co-hosts, so you can feel free to take it away. And feel free, folks, to put your questions either in the chat or we'll do Q&A at the end of the presentation. And raise hand, we'll do that by raising hands. But yeah, thank you.
Awesome. Thank you so much for the welcome. It's good to be back. We're excited to be here and share with you all our experience. I'm Mack and I use she, her pronouns. I'm going to be driving the slideshow here. So let me just pull this up. And I'll just dive right into it. All right. Can everybody see that okay?
Yeah, looks good.
Okay. Thanks. So, yeah, I'm Mack, and I've been working with Jade here at Metamimicry since we established in 2021. And we've primarily been focused on like backyard scale bioremediation. So that's what we're here to talk to you all about, our experience with that, where we see this kind of work going and how you can tap in. When we first started, we were really focused on like an on-the-ground experiment, and developed this mission sort of around that phase of our time where we were doing a backyard bioremediation experiment, literally in my backyard. So we really wanted to focus on exploring accessible biological solutions for our time's most pressing environmental pollutants. That's our mission. Since then, our like understanding of bioremediation as a whole has really expanded. So we now think of bioremediation as it relates to not just the environment, but also our body, our bodies, our culture. And then we've really leaned into this like access portion of our mission, the accessible part of our mission. So you'll hear us talk a lot about that as we go through today. So a lot of our work has involved some experiments, some on the ground work, but primarily at this time we've kind of shifted into like an educational space with bioremediation, and promoting access through that. Jade's got some exciting projects she's cooking up right now, so they'll tell you a little bit more about that later. But we got together in 2021, as I mentioned, and we were co-executive directors for a while, and I've since moved on to just being the chair of the organization now. So Jade is really driving the show, and I'm just supporting from like the back end of things, the board support side of things. So, Jade, did you want to add anything?
Just that I moved to Florida, so we have like a chapter now in Florida
Yeah.
and Washington. So yeah, across the US.
Yeah, we're bi-coastal.
Nice. Yeah, we want to always recognize our teachers, the lineage of knowledge that we're in. Lila Darwish, badass, wrote the like original grassroots book called Earth Repair. Highly recommend. And when we talked to her about it, she was like, yeah, I just wrote it because nobody else has written something like this. And she worked with other people to put it together, which was just really inspiring for us. It's like a lot of times I think people get caught up in having the imposter syndrome. Um, but with things like this, it's just like important to get in there and like be messy and learn. So she's super inspiring and knowledgeable. She does a really good earth activist class that I've taken twice. I also highly recommend that. Ava Arvest. Mack worked a lot with Ava at Myco Uprisal. That's a local Olympia mushroom cultivator. We learned a lot from Ava. Sal Rosales with Potter Valley Tribe. He's the tribal chairman. Really cool mushroom tribal leader. Yeah. Maya Elson. She also has roots they also have roots in Olympia and ended up starting Co-Renewal, which we'll talk about. Jess Ann Rubin is on the East Coast, the Northeast, has done work through Myco Evolve. Jess wrote this cool how-to guide on growing your own AMF, arbuscular mycorrhizal fungi. So you can check that out. It's like available for free. And then Daniel Stevenson has done a lot of work at the end of bioremediation, what to do with your contaminated substrate. And then Jay Schindler, I've done a intensive mycoremediation class with him. And yeah, he's also just very all about like expanding access for this type of thing. Super knowledgeable. Is that me too? So we'll talk more about the bioremediation network, but we've come up with this definition of bioremediation with Lila, and we have basically decided it's an umbrella term for allying with living organisms along with other regenerative tools to immobilize, extract, consume, and or break down toxins in soil and water in order to repair polluted ecosystems and support community health.
So bioremediation is a pretty, like, on its own, has a pretty expansive definition. So we want to be really clear about what we're talking about when we are talking about like backyard scale bioremediation. Um, the the scale that we tend to be talking about when we're talking about backyard bioremediation is in the in situ scale. And that means we're working on the site. This tends to be more grassroots bioremediation, but you can also have industrial scale bioremediation that's operating either in the site or ex situ. So it's like a scoop, basically scoop up the top like few inch layer of the soil, which is extremely expensive, take it to another site, and then deal with it off site. There's pros and cons to both, I would say. So when we're talking about the industrial scale, especially the ex situ kind of approach, where some of the pros include you're dealing with like a much more controlled environment. It's a lot faster. You tend to know what you can expect of the results. If you're dealing with this at an industrial scale, you have really good testing, so you can do a lot of pre-testing, post-testing throughout the process. But it also tends to be really expensive. That's why it's undertaken at the industrial scale, and tends to be applied for like really highly polluted sites. But if you're talking about dealing with a site that is less polluted, it might not make sense, just from like both a financial and like feasibility kind of scale to be treating this industrially. Nor do we see a lot of effort or attention and action like applied to this if we're dealing with lower concentrations. You can imagine like, there's perhaps like an EPA or Department of Ecology set limit for toxins in the soil or in a waterway. And if you're just a little bit, like a couple parts per billion or a couple parts per million below that limit, we might all know and be able to recognize like that's still polluted, but it falls below that limit for some reason. So that's not going to get prioritized at the industrial scale, but that might be a really great place for grassroots bioremediation to step in, or that's often where we see grassroots bioremediation stepping in. So when we're doing grassroots bioremediation, it tends to be on the site itself, in part because it's a lot cheaper. It's a lot more accessible for people. It's also less energy intensive, tends to be less disruptive to the environment as a whole. But some of the drawbacks are that it can be a lot slower. It's a lot less predictable in terms of the results. You don't often have a lot of that pre and post testing or ability to test throughout the process on the site. We're just dealing with very different technology between these sites. And of course, it's not as effective for those really high concentrations. So ideally, if you have a highly polluted site, you do have some kind of industrial scale intervention happening, but that's not always the case. So those are situations where grassroots bioremediation is actually really helpful. Some of the contaminants that we could be dealing with between these types of sites or across both of these scales are like the primary ones would be heavy metals and chemicals. And those have very different treatment approaches. So the whole time that we kind of talk about bioremediation, or personally, at least when I'm approaching a site or a situation, I'm like calculating, like, okay, what's going to be the best thing to apply here? First, I need to understand like how sensitive is this ecosystem? How deep does it go? Like, what's the scale of it? How bad is it? And then what are the contaminants? And that will help you identify like what your solutions are going to be at that site. Because heavy metals are going to be treated very differently than a contaminant that you could perhaps actually break down. Because heavy metals you can't break down, you can only take them up or immobilize them or kind of make them inert. Some other contaminants that we're dealing with include nutrients, radiation, or other like social or psychological or political kind of contaminants. And that's part of our expanded definition of bioremediation.
So we're going to walk through 4 different sections of the bioremediation organisms. Mycoremediation, myco meaning mushrooms, as y'all know. That's kind of our bread and butter. Me and Mack have mushroomy backgrounds. But yeah, mushrooms are a good thing to start with in this process. As this quote says, they're good at breaking down more complicated toxins into simpler ones for the microbes and others to deal with after. I think you guys know the difference between mycelium and mushroom, but the mycelium is the root network underground, and the mushroom is the fruit that pops up to spread spores. And both mycelium and mushrooms can do bioremediation. And yeah, the reason mushrooms are especially adept at breaking down toxins is because they don't bring the toxins into their body. They use enzymes to break it down on the outside. So that's why they're especially good at this stuff. Another cool thing about mushrooms and bioremediation is you can train them, aka playing God. You can precondition fungi to target specific pollutants by exposing them over time to contaminants in controlled settings. There's these people that we know that have dropped oil onto a petri dish and taken whatever mycelium has grown over that oil, and that part of the mycelium is more primed to deal with it. So that's really cool. You can also select and isolate certain species for the environment that you're in, which would be really good if you're trying to cultivate wild or outside. Yeah, when you're growing your own, that's something to consider is what's local. And you can think about what substrate to grow in. A lot of times mushrooms can grow on what we consider waste. So cultivating on a local waste substrate is ideal just to kind of get some of that out of the landfills and growing bioremediative organisms or food. One of my favorite things about mushrooms is that you can take the tiniest little piece and grow like 1,000,000 pounds, which is just very inspiring and conducive to this type of work. Arbuscular mycorrhizal fungi I mentioned before. Yeah, Jess Rubin has a how-to guide on how to grow them yourself. These are the type of fungi, they have a relationship with plant roots. They enhance nutrient uptake. They improve soil structure, increase plant resilience. They support biodiversity, and they don't have a fruiting body, so it's a little harder to identify them and to know what's going on down there. But they're incredibly useful organisms. Most plants do have a relationship with AMF, but there's certain ones that don't. The members of the Brassica family don't have a relationship and don't have AMF on their roots. And then we have fungi like trichoderma. So trichoderma is an interesting one because it is like the bane of mushroom cultivators. It's the green mold. It's a very common cultivation contaminant, but there's been studies that trichoderma can help degrade a variety of pollutants, including glyphosate. They can be really beneficial. Plant pathogen, or to reduce plant pathogens. They can also be anti other types of competing fungi. So yeah, don't just there's no waste in nature. So trichoderma is useful in bioremediation as well. And there's some considerations, again, with mycoremediation. If you're cultivating your own species, don't work with parasitic fungi or invasives as best you can. They're not ideal for removing or extracting metals from the soil. That's best left up to plants and other organisms. And yeah, like I said, we're starting with mushrooms because they're usually the best to start with in these kinds of bioremediation processes. So next up, we got the microbes. Oh wait, no, I gotta talk about, oh yeah, co-renewal. Co-renewal, I mentioned Maya Elson, started Olympia, went down to the Bay, started Bay Area applied mycology, went further south to South America, and they've been working to do oil cleanup in the Amazon. They did a lot of that. That's where I first learned about co-renewal. Super inspiring how they work to remove a lot of oil from the Amazon to do cleanup. And was also working with local people down there to grow food, like oyster mushrooms as food. So we were really inspired by them. Currently, they're doing post-wildfire remediation and have these really awesome projects in California. So yeah, they work with mushroom wattles. I think still oyster mushrooms primarily to do a lot of cleanup. So yeah.
Yeah, I'll just add to that. One of the things that they're really focusing on, maybe I'll go back, in California doing these post-wildfire remediation projects is that they're trying to incorporate more funding for testing, like pre and post testing, which is incredibly difficult to do when you can't predict a wildfire before it happens, like where it's going to be, what's going to be present. And so that pre and post testing, like you can think you might want to do pre-testing before the fire, pre-testing after the fire, but before you apply the remediation, and then after you apply it, and then at different periods of time afterwards. So that can get really expensive really fast, and it's difficult to have like really responsive funding, but they seem to be doing a pretty good job of like building up a name for themselves and a network to be able to get that. That's one of the major challenges that we see with grassroots bioremediation, is just that funding for testing is really scarce. And setting up like good experimental design is pretty difficult when you're working at these scales with people that are coming from all different kinds of backgrounds and maybe not just like a strictly scientific background. So that's something really important to keep in mind. Those are some major challenges that we see. There's lots of ways to overcome those challenges, and we'll kind of keep touching on those as we go along. But I will jump into talking about microbial remediation here. Um, as we said, bioremediation has this really expansive kind of application, and there's all these different tools within bioremediation that you can be working with. We tend to favor mushrooms. They're kind of like the poster child for bioremediation and grassroots bioremediation right now. They're really fun. People love to work with them, but there's a lot going on that we don't see as much of. And microbial remediation is one of them. It tends to be happening like at all times. Actually, bioremediation in general is happening like in all of our bodies, in every organism, at all times. Every living thing on Earth is doing active bioremediation. We have toxins in our bodies, things that are produced by our bodies that our bodies need to break down. So we have bioremediation pathways built into every living thing. Microbes are a really big part of this. We have like so many different ways we could talk about microbes, but because I'm working in wastewater right now, I'll just talk about wastewater as a little example, I think, of microbial remediation. So for starters, I'll say when we're talking about microbial remediation, we tend to talk about needing to apply microbes on a site, and they're not going to be good at taking up our pollutants and giving us like an easy body to extract and then bring elsewhere. So really, if we're talking about trying to treat heavy metals, we're just trying to make those heavy metals less reactive or to keep them in place. So they don't tend to be very good for dealing with heavy metals in general, because it's just going to leave them on the site. Where they do tend to be really good are working on transforming like nutrients or transforming chemicals into other components that will be less toxic or less reactive. Some of the ways that we do that are through, there's bioaugmentation, which is like essentially like feeding the microbes, giving them extra food sources, giving them extra stimulus, adding more microorganisms to sort of augment their capacity there. So you can like pull them out, grow them really big and strong, and then put them back in. And that's kind of a bioaugmentation approach. The biostimulation approach would be if you don't pull them out and put them back in and do that, you just kind of give them extra stimuli. This could be extra stimuli like heating the site. It could be extra food. It could be giving them things actually that they don't like to kind of get them activated, to get them excited, get them going. And then biostimulation could also be like giving them more oxygen. A lot of the times when we're dealing with microbial remediation, we're really just trying to change the chemical conditions at the site. So you want to change the oxygen levels, you want to change the nutrient levels, temperature, things like that. Those chemical and physical conditions are what you're trying to change to get the microbes more active and make them more effective. Compost is like a really easy way that people end up interacting with microbial remediation. At the same time, I told you I would talk about wastewater. In our bodies all the time, we got microorganisms doing microbial remediation in our guts, right? That all gets flushed down the toilet, or some of, most of it, I would say, gets flushed down the toilet and goes to sometimes a wastewater treatment facility or a septic system. In both these cases, we've got the microbes from our bodies are leaving, going to this other site, this ex situ remediation site, and the microbes from our bodies then are actually breaking down the pollutants that are in our wastewater. And wastewater treatment facilities are effectively farming through bioaugmentation the microbes from our bodies to help break down those pollutants further. So Prita mentioned the work that I'm doing now is working with Department of Ecology, working on the Puget Sound nutrient reduction grant program. We're dealing with excess nitrogen in the Puget Sound. And one of the ways that we deal with that is through bioremediation at wastewater treatment facilities, where we try to create different aerobic and anaerobic and anoxic environments to stimulate like denitrification processes. So this type of thing is happening all over the place. I am really excited to be nerding out about microbial remediation right now. And I hope that you think that is cool too. Um, Jade already mentioned um, co-renewal, formerly Amazon Micro-Renewal. They were actually applying microbial remediation, or they were working with this group who's applying microbial remediation. So they were out in Ecuador in the Amazon dealing with Chevron oil pits, applying micro-remediation, and then ended up that um, co-renewal. I keep like messing up their name because they changed it. Co-renewal came back to California, right? And Amisacho kind of built up out of that. And so they are working with biochar out there and inoculating biochar with the local microbes that are growing at these sites. So similar to what Jade described with putting a little bit of oil on a petri dish and seeing what fungi can grow on that and growing those out. Instead, they're basically taking a little bit of the microbes from a contaminated oil pit site, growing those out in compost and mixing that with biochar, and then applying that back on the site to effectively break down the oil pits that have been left behind by Amazon. And this is just one of those sort of like cases where I talked about industrial scale bioremediation being really necessary. But sometimes it's not always happening. Sometimes these companies can't be held liable to do the job. And so that's when grassroots bioremediation is stepping in. Even though this scale is really large and requires industrial scale bioremediation, this is an example where grassroots bioremediation is stepping in. Plants are a really important part of the picture here as well, and they have their own unique approach and pathway. And they're particularly useful, I think, for heavy metals, in part because when you're growing a plant, you can suck up heavy metals into the body of that organism, and then you can pull out the plant. And now you have concentrated heavy metals in that plant. Of course, that's important to consider when you're thinking about eating these things or what you're going to do with that plant after the fact. But there's a lot of different pathways that plants can take. They can really help stabilize the soil, prime it for other things to go in. But yeah, my favorite one would just be the extracting heavy metals there and concentrating them. Different plants have different capacity to do those things. Some plants make really bad bioremediators for heavy metals. They can hardly get, like, for example, something really heavy like lead, they can hardly get the lead up into their roots. Other plants, like members of the cucurbita family, that's like pumpkins and cucumber, or like sunflowers, are really good at pulling up those heavy metals all the way up into their body and like getting them into the fruiting body. So it makes it really easy for us as bioremediators to go in and pull those out and then figure out how to treat it elsewhere. So we see groups like Rise A Renewal Collective, they're working out in Baltimore. And this is, I think, one of the like really classic examples of where grassroots bioremediation is really applicable. They've got abandoned lots there that are highly polluted with heavy metals, especially lead, and they've been applying sunflowers, which on their own are already kind of like a symbol of hope. But they've been applying sunflowers to suck up those heavy metals, and they've been doing pre and post testing there. And it's been really effective for bringing more support as well. And so that's one of those places where bioremediation touches like these other aspects. It's not just like what's happening in the soil, it's also bringing people from the neighborhoods together and changing their attitudes about the sites that are in their neighborhood. And I believe since then they've been able to start growing food on the soils because they've effectively lowered the contaminant levels so that it's now safe to grow food in those soils, which is a really big deal to be able to turn like an abandoned, polluted lot into a community garden.
Worms. Worms are awesome. So are several other animals that are kind of the key bioremediators. Like Mack mentioned, everything can bioremediate, is bioremediating, but there's some special guys out there. Yeah, I learned actually this. So worm remediation is like worm remediation with worms, but if we're looking at all animals, it's called zoo remediation. Um, Worms are engineers. They're engineering in the soil. They're digging these pathways. They're aggregating. They're aerating. So they're doing a lot all the time under the earth. And yeah, their castings feed bacteria and they improve soil fertility. They are awesome bioremediators. And then we have aquatic zoo remediation. I've been learning more about this down here in Florida. I'll talk about them, but oysters are super primed to be biofilters. They can remove nitrogen, phosphorus, sediment, and heavy metals from the water. One oyster can filter up to 50 gallons of water per day, which is crazy. But there's other aquatic animals that bioremediate as well. There's oysters, there's mussels, crabs, lobster, certain fish, and certain marine animals. And then we have the terrestrial invertebrates, like the roly-poly. I think this is a Latin name, Armadillidium vulgare. Roly-polies have been shown to accumulate heavy metals in their bodies. They can be harvested to remove contaminants from the soil. There's a little bit of an ethical quandary with that. You know, if you're working with animals, kind of using them to get heavy metals out of the soil and then just killing them. I don't know. There's a little bit of something to think about in that. And then there's also beavers, which are really cool ecosystem engineers as well. Beavers act as bioremediators because they basically create wetlands that are nature's water filtration systems. They slow down water, allowing pollutants to bind to sediment, and they improve the water quality downstream. Mack is a beaver fan. And then you can't forget about us, can you? Anthro-remediation is a made-up word that we made up. But so we create the pollution. So in a way, like reduce, reuse, recycle in that order, we can use our human power to reduce the types of pollutants we create, take responsibility for our impact. We also have the gift of mobility in our human bodies to like grow a mushroom here and then put it in the place where it can bioremediate more effectively. And we can also develop systems and use our human minds to create better functioning systems that work better with the natural ones, the pre-existing natural ones. So yeah, can't forget about us. And then our example is ore. So they're down here I'm in Sarasota in Florida. This guy Damon Moore developed this thing called an ORP, an oyster rag pot, which is a very simple, low-tech design to grow, to house oysters. And they've been doing this really big project in a big river here, and it's been very successful. One thing that I took away from Damon when I was talking to him is that he took a long time to get to know like the people, the ecosystem, the players, like years and years, and did a lot of studies on what would be the cheapest and most effective like low-tech design and came up with the ORP. And so something like he took his time to figure out what would be really effective. And I think that lends itself to their success, and something to remind us, slow down. So once you have your organism, then you have your installation method that you need to think about. Where are you going to plug them in? Like I mentioned, Damon created the ORP. There's another oyster remediation group here. They've created something called a VOG, a vertical oyster garden, which is basically just rope, old oyster shells on a rope, which can house baby oysters, and they suspend them under docks. So those are two installation methods. Compost tea. Compost tea is made by steeping compost in water, and it makes it more spreadable because you can just, spray over a wide area. There's spore slurries, which are kind of similar to compost tea, except you're working with mushrooms, and its spores are mycelium suspended in water. Me and Mack were once messing around with a spore slurry squirt gun to try to launch the spores over the land. And then there's biochar, which Mack talked about with Amisacho. Biochar was actually inspired by Terra Preta, which is this way of intentional soil management by indigenous Amazonian people. Basically, what was found was that the place where they've layered all their waste and even like the dead, like compacted the soil and made this very carbon rich soil, which becomes an incredible amount of like holes and space and surface area for microbes to be housed. Another way I like to think about these things is like we're basically trying to create the best home for them to live on. So biochar is a hot topic too. And one thing that's cool is that you can use another waste product. Like when I worked in California, they were culling a lot of trees for forest fire management, and the tribe I was working with was turning those cold trees into biochar to then respread over the land after being inoculated with certain microbes. Wattles, that's been one of my more recent obsessions, especially for mycoremediation. Like we mentioned, in California, they're using wattles for post-fire remediation. Wattle, like you guys have seen wattles by construction sites. They're these burlap snake-looking things that are used to catch erosion, but they're usually filled with straw. And some smarty pants was like, oh, well, let's inoculate that straw with fungi, and then it goes from just erosion control to also a filter for any water running through it. So that's really cool. Then there's bunker bags, which is a similar idea, but an actual bag. And I've heard people use them on the edge of lakes or even floating in bodies of water or even burying them in the ground to absorb some of the pollutants. Bioswales. I'm sure you've seen bioswales as well. These are often in urban areas. It's like where the land is swaled. And there's a lot of, Max circling that. There's plants and biology that line the swale and help filter and slow down the water before it reaches the water table and waterways. Bioswales can use all types of different bioremediators. And I'll talk about that more. That's what I've been focusing on in Florida. Bioretention, similar to bioswales, but they look more like ponds. They also, they look more like ponds, and it's not like a channel for water. It's just a place for runoff to collect and for microbes and things to slowly break down the pollutants. And then there's, I don't have it in here, but floating islands. Those consist of a raft or platform that is on a surface of water, and it supports the growth of wetland plants. Those can be a cool way to get bioremediation into a body of water as well.
I would add that those tend to be more effective actually in bioretention settings or like detention ponds. Just
Oh, cool.
so the dual application there is like a lot more effective than just launching it into a lake, for example.
That's cool. Oh, well, wait, what if I have a wattle that is soaked with stinky pollutants? What am I supposed to do with that?
Gosh, that's a really good question, Jade. Um, this is a question that we get a lot, and there aren't really great answers to this question yet. When we're dealing with remediation, often we're dealing with like really highly polluted sites that are presenting a problem to our health, to the health of the ecosystem. And so we need to do something like to address that at that site pretty soon. Um, but then you have this problem after the fact where you've got now like a sack of pollution and you don't really know what to do with it, especially if it's like heavy metals. You can't break those down, so you're just kind of stuck with it. So some of the approaches that people take are like putting them in a cell. We do this with like landfills, for example, or like we do this with nuclear waste. You kind of just want to put it in a cell somewhere and like make sure it can't leach back out into the environment. If it's something where the levels aren't too high or they're more like chemical compounds or something where it's not heavy metals, you could just add more compost and continue to break that down, kind of dilute the pollution essentially, and then you can spread that out elsewhere in a manner which isn't going to just reintroduce like high levels of pollution. Um, there's kind of like recapturing and repurposing, those go together a little bit. We can actually, like, I've heard of, I don't think it's very cost effective, but we could like take mushrooms, for example, that we have uptaken heavy metals into and do an acid extraction of the heavy metals in those. And so we can like t reclaim those heavy metals and then repurpose them. Doesn't seem super feasible yet, but people are working on it. The landfill is kind of it's basically putting it in a cell, but at least those sites, we kind of know what's going on there. They are usually looked after, and people are like treating them very carefully to make sure the contaminants from those sites don't go elsewhere. Ideally, you're taking it to like not just the landfill, but the Hazo house at the landfill because they know how to address that appropriately. And it's really important to consider that like these, like whatever method you choose has the potential to really impact our food web and to like reintroduce contamination to these ecosystems and sites. So you want to be very intentional about the way you go about it. Um, for example, if you just grew a bunch of sunflowers, but you never harvested them, you might temporarily make those heavy metals inert or remove them from the soil, but eventually, if you just leave that site the way it is, they're going to go back. So making sure that you have the time to commit to kind of see that full life cycle through is really important. Or the birds could be then eating those seeds and bringing them elsewhere, which could be good for diluting the problem, or it could be a problem for the birds if they're consuming too many. Um, to date, we don't really have a great answer. Jade mentioned Danielle Stevenson out in California, who is kind of doing a lot of work to figure out answers to these questions. But no silver bullet there. One thing to really remember when you are participating in bioremediation as a bioremediator organism yourself is that you are sensitive to these things. That's why we're dealing with them. And so we really need to take care of ourselves as we're doing that. That means limiting our exposure, which primarily, your first line of defense when you're limiting exposure is to like not touch it, not breathe it in, not be there as much, but you also need to be there if you're doing the work. So if you can wear the right gear, um, like you might need a respirator, gloves, pulling your hair back, covering your hair, having like specialty clothing that you're wearing and taking that off. This is all going to be really dependent on the site that you're working on, and you need to do your research to know what you're participating in and what the risk is to you when you're doing this work at the site. But some other really helpful things to consider as well are just that like your body, as we've talked about, is doing bioremediation all the time. So you can just support it in that process by staying hydrated, participating in these activities with people, staying connected, um, finding joy throughout it. If you have a really negative attitude and you just feel like you have to solve all the world's problems with this bioremediation project that you're doing, you're not probably going to be taking care of your body very well. And then you're weakening you as a bioremediation organism, and limiting your ability to do that work. And actually, a lot of these bioremediation organisms that are helpful on the ground are also helpful in our bodies. I'm actually these days a lot more interested in mycoremediation, like internally, and so more of like consuming mushrooms for health, alongside doing the on the ground work that people are doing.
I'm gonna just breeze through this slide because we'll talk about examples. So this is basically like if you're personally interested in starting your own um, project, these are the steps you can take that most people have done. We got a lot of this from Lila Darwish. But yeah, organize your team. Make sure to include people with skill diversity so you can touch all the bases. Look into the ownership and site history. Ask neighbors. Look for lusts, leaking underground storage tanks, past land use records. Find funding and donations. We've had struggle with this because not a lot of people know what like bioremediation means really, and when you do, it's kind of on this like huge scale. So that's why a lot of times we work on with like growing food because people are like, okay, that makes more sense, and we can sneak in our bioremediation along the way. But yeah, doing that. I think in Washington, it's more there's more funding available for that kind of thing in California too. Um, Conduct the site assessment. This is understanding the land. Look for whatever's around, like whatever fungi, plants, microbes pre-exist. On one of the sites, I found like a local turkey tail, or a turkey tail on the site, so I used that to inoculate the wattles. Soil and water testing. Make sure to check for free programs or for reduced cost because there often is, and also be specific about what contaminants you're testing for because there's a spectrum of pollutants within certain categories as well. Determine a remediation treatment plan. Yeah, how will you know if you're successful? Think about that. Matching specific organisms to specific contaminants. No one size fits all. Remember that. Depends where you are. Think about the seasons and things. Engage with people. Do an initial site cleanup. Sourcing materials. Thinking about where you're going to get your mushroom spawn, your compost materials. And yeah, maintain community engagement and then test for the results. So you can compare your results to the baselines. Did contamination levels drop? By how much? Document what worked, what didn't, and then share learnings. This whole list, I'll talk about the bioremediation network, but we're trying to like make this easier and have a place to centralize these kinds of projects so we can all learn from each other. Because doing this stuff, you notice that we're all just kind of in the dirt and we're not like connecting, and it'd just be really helpful. Yeah.
I just want to acknowledge I'm seeing some questions in the chat. We're going to get through like some of the work that we've actually done, and then we will have hopefully time to address those at the end. So, but they are good questions coming through. Yeah, thanks for setting up like kind of that process, Jade. We've just gone through like a lot of background information, and some of it I think has been sounding a little bit vague. So now we're going to talk about what we've actually been doing. When we first started we formed because we heard about the discovery of the tire chemical transformation product, which is known as 6-PPD-quinone, which is killing off our coho salmon in urban streams. We started noticing symptoms of urban runoff mortality syndrome in the 90s, which was killing up to 90% of the coho salmon, in some places 100% actually, as they were returning to spawn in really urbanized streams. And was this phenomenon was starting to be noticed in the 90s, and then it actually got worse as restoration was starting to take place in really heavily urbanized areas, or not quite restoration, but like recreating access. So, like removing culverts or barriers to upstream migration for coho. So then they could return to these streams, but they were in heavily urbanized settings and they were just being killed in the waters that were the same waters that were triggering them to come back upstream. So really terrible phenomenon. I'm glad we discovered it. In my undergrad, I was in class with some folks who were actually working on that project, so I was a little bit aware of it when it came out, working at a mushroom farm, and was really excited to try to find a solution, see if I could blend my chemistry background with the mushroom cultivation skills I'd been learning. And around that time, I met Jade. And so we went through the literature, identified some similar studies that had been done before we knew what 6-PPD quinone was to try to treat stormwater. They were using bioretention cells. So they were basically 55-gallon drums with like a layer of gravel and a sand compost mixture, and then topped with a couple inches of wood chips. That's a pretty standard, like best management practice at the state level for treating stormwater. And some of the studies already indicated before we knew what the chemical was that if you just treated it that way, it would reduce the mortality rate for these salmon. So we'd seen that they had done a couple studies where they tried to inoculate like the top layer with a fungal amendment, but for a couple of reasons, those experiments didn't really end up working out. Like people dropped out of the experiments, things like that. So we thought that would be a really good place to jump in. And we did more of a pilot scale project. This was literally in my backyard. We inoculated these cells with different types of fungi, and just kind of reduced the scale that we were at just to see, like, is this feasible? Is this something we should really pursue at a large scale? Because when we originally did the calculations, it would be like, like a $350,000, $400,000 project if we were to do it like the full scale that we wanted to. So we scaled it back, and I think we did it for like $15,000 or something, maybe even 10. We got really scrappy. We got some deals from labs and lots of donations and a lot of support to make this happen. We did have some of the cells die, which kind of shows you, well, maybe this isn't like really an effective intervention long term. And that's what had been happening in some of the other experiments as well. So there's one thing. The other thing was that while we didn't have, because of that, we didn't really have enough data to support like a robust statistical analysis for this. We could kind of piece together some things to identify that the cells that had fungal inoculants did not perform as well as the cells that had no fungal inoculants, which we know that there's microbes really active in those cells that don't have the fungal inoculants. So we could kind of think about it as like, well, fungi versus microbial intervention here. And those cells actually reduce the 6-PPD quinone levels pretty significantly as compared to the ones with active mycelium growing in them. The stormwater that we collected was from a detention pond, and so the level that we were seeing in it was already below the median lethal level, which was good. But we know we don't want to have like any level of it. Median still means that a certain amount of salmon are going to be dying from this chemical. And now we know that other fish are affected as well. But with those findings, we decided not to pursue a $350,000 project. And this was a really important learning lesson for us, which is to say that fungi are not always the solution. And you really do need to know what you're applying. You need to do your research. You need to have testing to back it up. We see a lot of people just saying, if you put mushrooms at that site, it will fix it, but it will not. It will not always, I should say. So that was a really big learning lesson. And then we started to think about, well, gosh, it was like kind of difficult to get the spawn in the first place and to get this whole project going. And so we wanted to focus more on the access side of our mission. And that's when we approached the Squaxin Island Tribe's Salish Roots Farm. It's their community garden to see if they wanted to have a community access mushroom program there. Now, our first interest in this was for bioremediation, right? And when we were talking to their tribal program manager, she was really interested in the bioremediation aspect as well, but their community had a lot of other interests that went really beyond bioremediation, and there were a lot of interests that related to food and medicine, and helping bulk up the soil in their garden and other types of ways of interacting with mushrooms. So it became a really like multi-purpose program. We developed a plan and then just built out the program with their needs in mind, and also understanding that this wasn't just about growing mushrooms for food and medicine, but it was also about building capacity and resilience in the community to address issues as they came up in the future. And once people had more skills and familiarity and knowledge, now we intentionally kind of like planned ourselves out of the program because we were working in a tribal community that wasn't our own community. We wanted to make sure that if we were going to be building a program, it was something that would be fully integrated into that community and they could continue to operate without us there, and that they would want to continue to operate without us there as well. And so we were there for, I want to say, a little over a year, probably closer to 2 years, but a year really solid, building the program up, creating all these potential pathways for them to run with it and training folks, and then taking that in the direction that they wanted to go. And it has, of course, changed since we left, which is good. That means it's being, picked up and integrated in the ways that they find most meaningful. And now one of the things that they're planning to do there is for the spore program. I don't even think I said the name of it. That's our spore program. The Salish Place of Remediation Education, is they're planning for that to be like a hub for other tribal gardens that want to grow mushrooms. They'll grow out the spawn at that site and then share those with other tribal gardens and build out like a larger network. So it's really cool, and I'm glad that there's still some plans on the table. And I just want to emphasize that when we were doing this project, we started with an idea and we had a general interest in building up a community access mushroom program, but that kind of integration takes a lot of time to figure out what it's going to be, who's going to interact with it, how they want to interact with it. And because we didn't know those things from the start, and because people didn't already have a lot of familiarity with mushrooms, we needed to approach it from this angle that was like, okay, really multifaceted, and also allowed for a lot of adaptability and for things to change with time. And the program is still changing and taking shape. So some of the ways that we approached this program were through workshops. We taught workshops on mushroom cultivation, both in like lab setting and outdoor, like log cultivation or growing just in mulch in the garden. We provided food for free that was grown there. I mean, they really provided it. They provided that food for free to their community members, which they give out at their farm stand weekly throughout the season. We also took folks on foraging walks and taught mushroom ID. We taught kids. We made food, or we made recipes, like we did cooking classes. We did medicine making classes. And of course, we'd feed people with these things and then take the medicine and like bring it to the elder center, things like that, to get this stuff integrated in like very different ways to get people interacting with the mushrooms and thinking about how they wanted to relate to them. Really robust project. I was like, I haven't been involved in thinking about this project for a while, and I was going through all my pictures this morning and just like, whoa, we really did a lot. We tackled this from a lot of different angles. You can see here, like Jade was working with um, some of the guys there out at the farm. We actually had to build the lab space. There was originally a plan for them to build a lab space. It wasn't working out on the timeline that we needed for our grant program, so we were like, okay, we'll figure out how to do it. Really, Jade figured out how to do it. Um, so we actually had to build a lab from the ground up. Before that lab, we had a pop-up tent in the barn. We had multiple versions of fruiting spaces, including like a chicken tractor we used at one point after the greenhouse had a like glass explosion. All different kinds of things were happening there. So we just had to be really resilient and take our time with the project and ensure that we were teaching people about what we were doing. And I think I just want to get to that as like that's the most important part of this program that we built up, and that's why I think we're so focused on teaching and education now. It's because when we built out this program, it was pretty clear that if we walked away, it would cease to exist. So Jade was really busy making all kinds of instructional videos on how to cultivate mushrooms in that lab, in their labs, so that they could see like, oh, this is exactly where I grab this thing. This is exactly like the button I turn on to turn on the flow hood, things like that. We also wrote a manual essentially for everything that we did at the program, like how to make a concentrated like mushroom extract tea, so that people could just steep it really quick instead of simmering for a few hours, things like that. To also how to cultivate, how to think about these things, and overall, blast, but wouldn't have been possible without teaching people all these things. One of the last components of this after we got all these things kind of set up was to train the people that worked there. And then also we brought on some interns, and that was really critical because one of our interns that was operating out of the space has continued to show up, and he's volunteering out there now. And then since I think got hired, is that right, Jade? He got hired out there to operate out the space, so that was really critical part of making sure that this space would continue to exist without us because we didn't need to be necessary in that kind of environment. So, and that I think ties really well into thinking about culture, mushroom cultures in tribal environments.
Yeah, we're running out of time, and
Sorry.
I no, it's all good. This is a part of
I think it's important, Jay, so you can go ahead and like, yeah, present this piece and then we can, yeah, it's fine.
okay.
Yeah, we can go over by a few minutes, it's fine. Mhm.
A lot of things too. This is a lot of what I wrote here and wanted to talk about is in the zine that we put together. That's I can share with you. I don't know if I have before, but yeah, it kind of lays out different ways that we can support Indigenous sovereignty through mycology. I worked for a couple of years in an environmental department for a tribe, and just learned a lot about, what's important to a lot of tribal entities, and just where my passion is mycology. It was wanting to see how I can better support Indigenous sovereignty. With the spore, that's a good example of food sovereignty, and food sovereignty initiatives. Mushrooms are really great additions to food sovereignty programs because they're a closed loop crop. They can grow on waste, and the waste that they produce helps grow more food, like more plants, and feed animals. So adding it into food sovereignty program is wise, in my opinion. And they're really rarely cultivated on tribal lands, so that's something that should be encouraged, and I think is more so. And then, yeah, with land management planning, the Karuk tribe in California, they've included the tan oak, aka the matsutake mushroom, into their land management plan as forest health indicators. And this is important to illustrate mushrooms as important for tribal uses, and getting more access to land to harvest mushrooms helps with land back initiatives, and is just another reason to support the land back movement and biodiversity on tribal lands. And what else did I want to say? Oh, intellectual property rights. So I think you guys have talked about Maria Sabina, which was a huge issue, obviously, with psychedelic mushrooms and bioprospecting and piracy. And something really important is making it clear that Indigenous communities have been working with mushrooms forever. There's this big, like, reoccurring thing with people we talk to that, like, Native people didn't use mushrooms, or like it's not a part of their culture, which is just not true. You know, a lot of this information is kept hidden because they don't want people to steal, the colonizers to steal from them, as has been the case many times. So creating these, like, being very mindful about they're like pre-existing protocols, and I'm really interested in like things that trace ethnomycological knowledge. As far as psychedelics goes, I don't know if you guys have talked about Chacruna. It's a really cool organization, which is Indigenous led. They have this Indigenous reciprocity initiative. So people, Western practitioners of psychedelic medicine, can pay like a cut of whatever they make, and it goes back to the original knowledge keepers. But things like that, like not just with psychedelics, but with all types of medicine, if that were included more, that type of thinking is where the knowledge actually originated, and how we know what's good for what, and things like that. So we worked with the Heron's Nest, which is a group up in Seattle who work with the Duwamish people. And then Yehao Show, they're the Indigenous Creatives Collective. They're a group of like urban native folk who also have this hub where they're trying to grow food and be like a center for people. And I think, bringing bioremediation to those places primarily, like first, is really important, just because of like cultural issues, like, you know, that cultural mediation that needs to happen. And also just they're like these places, these like community places where people gather, and like making those places, the land healthier, is really important to focus on. I think, and then next, I just wanted to, Mac, would you hit the next button? The next, something we've been working on coming out of Lila Darwish's Earth Activist program last year, there's a small group of us that are working on the bioremediation network, which is kind of distilling all of these, all of this knowledge and putting it into a centralized place. We also are working on refiguring out the definition of bioremediation, as it's a like ambiguous term kind of right now. We want to make sure that it stays in the right mind, the right way. So we're looking for people to help volunteer, and we would love if any of you are personally interested. I think it's an amazing project and initiative. We're right now looking for web development support, and then people who want to outreach to folks to find bioremediation projects. We have a survey they can fill out. We're just now populating the map. So I'm super excited about that. My favorite thing about the bioremediation network, and perhaps bioremediation in general, is this inspiration. It's like, in these times that we're in, it's a remembering and like a visual, the map is like a visual representation of all these people around the world who are working on these projects and doing it low key, but like still doing the work. So yeah. And
Do you want me to click the link or maybe um
you guys can click around. The website is bioremediate.net, and then the map is on there too.
I'm typing away furiously in the chat, so I'm trying to get to questions as I see them, but apologies if I've missed some.
It's fine. Yeah, I think folks can, yeah, that's fine. They both can just click on the link on their
Cool.
own. Yeah, thank you.
I think we're almost done too. I just in Florida, I'm working on. I'm still getting to know like who to work with, connect with plant people, but basically wanting to work in bioswales because flooding is a huge issue here, obviously. Um, so yeah, learning where to plug in that way.
I will just leave you with these words of wisdom here. Um, I say this because something about the mushroom realm attracts a certain type of person sometimes, or a certain personality type. I don't want to pin it on like individuals, not like the people themselves are bad, right? But there can be these like sort of toxic ways of approaching this work. I see this a lot as like elitism that comes out. People are like, oh, well, I know the scientific name of this mushroom and you don't. So that means like I know everything about mushrooms and you don't. But really, this is such a broad world and there's so much to interact with and learn about in it. And we all have so much to learn from each other. I think for me, just being humble throughout this process has been huge because Jade and I undertook like a really big, robust project with the Spore program, and have continued working on other projects, but never, I don't think at any time during that process did I feel like I should be the person doing that. It's just that nobody else was doing it, and we wanted to see it happen. So it was a really big lesson in humility to like show up and just try and to learn from other people. And I think that was really cool because people were able to share stories with us and share knowledge and course correct. And like I've seen some questions in the chat that are, I think, maybe attempting to course correct too. And I I welcome that, and I think that that's the right attitude to have in these spaces. Is not like trying to show up and say, "Well, I know everything," but really just to come in and come correct and learn from each other. And that's why we wanted to be here because y'all are here learning from each other, and we're here to learn from you too. So thanks for having us. Thanks for sharing in this conversation. And if there's time for questions, we can take them. I didn't put a slide on here with my email, I realized, but I will drop my email in the chat. Jade, can I drop yours as well?
Yes.
Okay, cool. And definitely reach out to us if y'all have questions. I'm really happy to talk about this. Um, I'm also on LinkedIn, which is kind of a weird way to interact, but you can find me there if you want.
Yes, yeah, thank you. Back at JTF, if folks can give some words of thanks for them in the chat too, especially let me see. Yeah, I think I mean, do y'all have a little bit of time? We've spent maybe like a few minutes.
Yeah.
I think so. Okay, yeah. So
Yeah.
a couple of things I'd like to. Well, one, I just wanted to uplift a few things you said that connect well to our themes, especially this week, but also what we've been studying over the quarter. And then also to my students too, if you can give some words of thanks. I see there's words coming in the chat. That's just good. And just do a give back. So just if you could also share a more sort of like generous kind of words of thanks, just recognizing Jade and Mack for something you learned from them. That would be nice too. So you can start doing that in the chat, and then I'll collect the your words too. I'll save it, and I can share it with them in a thank you card. So you just do your give backs while we're wrapping up too. That'd be great. And then I'll say a few things, and then I know you've been answering some of the questions in the chat, but maybe there's a couple I can just read real quick. Um, And so, yeah, just wanted to uplift a few things. I think, um, yeah, this week we've been making connections with social movements and how we can kind of learn from um, mycelium networks in terms of how we like as people organize our social movements, kind of similar in like the emergent strategy of Adrian Brown, kind of in that vein. And so, just some of the points y'all made, I think it reminded me of some teachings of like the Gilda Shepherd from the Tacoma campus. One of our colleagues there said that yeah, some of the like animations were funny. You know, we're in a rush, so that's why we go slow. When she was making her film, since I've been down, she does a lot of work, or yeah, she's done a lot of work in the Washington State prisons doing education, with incarcerated folks. And when she was making the film, she said that was like their process. You know, we're in a rush, so that's why we go slow. And so I think that's like that came through and both like the way that you work and some of the other organizations that you talked about as well. And I think that that challenges some of the we talk about like white supremacy culture, like this culture of urgency and stuff like that. That's is a challenge to that. I think the work both that Metamimicry does and then the different organizations that you cited to that you talked about in your presentation um, represent this work called like anti-colonizing science, and that's in the words of Max LeBron. But we've talked about that in our discussion boards and in some of my recorded lectures. And so, yeah, just great examples of that. 'Cause I know many of our students are they're learning science right now. You know, they're learning like some of the mushroom identification and some kind of foundations of mycology um, this quarter, and some of them are going on to do more upper division science work, especially in their certificate. And so having this model of how to do science in a way that is liberating and yeah, anti-colonizing, I think is powerful. So thanks for giving like a real world example of that. This idea of like working yourself out of a job is an important one too. So I think like what you did with the Spore project was sort of an example of that. When we're working with communities, it's like the sharing of knowledge and being transparent and passing on knowledge is really important. It's a way to challenge the elitism too, I talked about. So knowledge doesn't get concentrated in the hands of like the experts, but gets like passed on to the people and really like centering what the needs of the community are in developing the programs and stuff. So the fact that y'all are open to that, like not just doing the bioremediation part, but then integrating some of the food production too. And then, yeah, relationship building is really key as well. So that's part of the going slow part to go fast. But the Adrian Brown also in emergent strategy talks about the organizing at the speed of trust, and so that's also I think a key part of that this work as well that came through in some of the groups you cited. Also with the way that you're doing science too, I think it's a great way that you're talking about the humility piece. Is a great way to end because in the workshop we did earlier, looking at the ethics of psilocybin research and development, we talked about obviously like yeah this notion of cultural appropriation and the biopiracy you talked about. We dove into like the sort of hidden histories and the harder truths about like the encounters between uh, the R. Gordon Wasson and Maria Sabina, and so some of the stuff that gets kind of whitewashed out of the mainstream kind of mushroom histories, and so the cultural humility like being like an alternative way to a framework an antidote to like the like extractive way of interacting with communities, especially indigenous communities. I think that was an important piece too that y'all really illustrated in your presentation. And I think just offering a lot of like inspiration anytime people feel really heavy right now with everything happening in the world. So I think, just offering a lot of words of inspiration is really important. And okay, great. I see some give backs coming in the chat. So that's great. I'll definitely be sure to save those to share with our.
Thank you so much for
Yeah.
having us. And this is like the third year I think we've done this. So you're the best. And if anyone wants to join the bioremediation network, you don't need to know about bioremediation, but just let me know. You can email me. I can get you in on it.
Oh, yeah, thank you. And maybe like a couple. Oh, I see you kind of responded to this 3D printer question, just scrolling through the chat. And then the Angelina showed to the a fungi that degrades PETG, looking into fungal degradation of plastic, anything.
I'm not familiar with that chemical or compound, but I'm happy to do a little digging myself, I can nerd out about that, obviously. So,
Mhm.
yeah, I think as a starting point, it's just good to get the kind of like the foundations and the basics. But primary lit is where I go because oftentimes people have already done like a little bit of digging. Um, if you have any kind of chemistry inklings or chemistry background, it can be really helpful to look at similar types of compounds and to try to understand those differences. And um, yeah, similar compounds can then behave similarly. It's not always the case, but it can help you kind of try to line up the right type of species. So yeah,
Mhm.
but send me an email and I can try to look into it.
Okay, cool. Yeah, and then there's also like the P, like this might be a different compound, but the PFAS, those apparently can't be broken down at all. Is that true? Like, but do you think there's any possibility that mushrooms in the future maybe could, or
I don't know if mushrooms are the future for PFAS. I actually work with somebody whose like husband has been doing this like PFAS project that we are starting to see some like remediation projects for PFAS come out.
okay.
But the scale,
There's one for
what was it?
hemp, I think, or working with hemp.
For PFAS?
Yeah, it was in the last Earth activist class. There's a tribe, I think, on the East Coast looking into it.
Oh, cool. Okay. Yeah, so there's stuff out there, but this one's kind of a newer, bigger, scarier one, so there's less info.
Yeah.
Yeah.
Cool.
Primary
Okay,
lit,
great.
great place to go.
Nice. Nice. And maybe maybe one little brief question in case like I see you've been responding in the chat, but if there's anything mind. And then I do want to like have at least one give back that like a student can share too. But yeah, I don't know if you have anything more to add to Moshe's question, Mac, about the toxicology, rather than I see his comment interested in this, Dr.
Well,
Dan.
and maybe something else, because that may be more difficult to answer within that context, is actually what, in your experience, is done with those fruiting bodies, whether it's plants or how do you actually, in your work, uh, manage the resulting polluted, whether it's, oysters in the water or sunflowers or whatever.
Yeah, that's a great question. That so we haven't done that much like on the groundwork. We've mostly focused on the education side of things. What I see is that people will take it to a hazardous waste facility or a landfill. I saw your question about if you're in like a really rural community or a place where you don't have access to like those types of sites. That's when like literally just getting some kind of container that's not gonna like leak, that you could bury is something that people do, or just storing like elevated off the ground so water can't get into it, so it can't leak. That we don't have great solutions right now. And I did see your comment too about like, well, you don't want to just be putting low level contamination back into the environment, right? It depends on what you're working with, is what I would say. Because like, for example, some of the time we're dealing with, like in the case of oyster remediation, oyster texture, um, we're dealing with excess nutrients.
Sure.
So there's not really harm in, right, like holding those nutrients in a
Right,
little.
or even glycophosates, you probably aren't that worried because they do break through that cycle. But I'm talking
Yeah.
about heavy metals and polluted.
Yeah, yeah. And so for those, it's like you kind of have to weigh like this is where it gets fuzzy in the grassroots realm because we don't often have funding for testing, so you know exactly how polluted it is, right? So I always just suggest erring on the side of caution and taking it to a hazard house if you have that access. Otherwise, storing it in some kind of container that will keep it safe and not leak and not get rainwater in it, that kind of thing, until you do have access to a place to store it.
And in your experience, those communities like in Amazon, etc., actually do that, or the temptation to then, just go and throw it somewhere in the backyard, right, or sell it to someone is too high.
That actually the Amazon one is a really good, I think, example of how people navigate like the food thing and the all
Right.
of that. So what they've realized is that the the mycoremediation piece is actually really difficult for them, like on a feasibility level. And so they're really focusing on the microbial remediation. So you don't have to worry about people like harvesting the microbes to sell those, right?
Yeah, for
They
sure.
also grow and sell mushrooms for medicine. They grow those on clean substrate. They're not growing that on polluted
No, for
substrate.
sure. But that's a different,
Yeah.
yeah, that's a
Yeah,
different
but it's
story.
very different. And there, yes, there's heavy metals, especially like mercury in oil, but they're primarily concerned with the oil itself. And so they're really weighing, and of course, that's not an ideal situation. That's like should be an industrial scale remediation site, but Chevron is like not willing to do what they've been told they need to do. So it's not ideal. There aren't ideal solutions for it, is all I can say to that.
All right, thank you. Thank you, Moshi, for that. I
Yeah,
think,
thank you.
I'd like to have one give back because I know we also need to take a break soon. We have a few more things to do in class, including, hearing Moshi's presentation. But I wanted to see, thank y'all for sharing your give backs in the chat. And with one of you like to read your give back, I was gonna just, well, I told Jess to read your give back if you don't mind. um, or you could just say it verbally. Jess is still here, right?
Yeah.
Yeah.
Hi.
Okay. Thank you.
I'm sorry. I'm also getting ready for a wedding right now. But I did want to say,
Congratulations.
in my experience in community projects, it's really easy to feel like you have to take all of it on. And I like that you talked about that in your presentation and that you have um, experience, both from a tribal sovereignty perspective and also just from a general community organizing perspective of passing your skills and knowledge and projects on to more people. And also just like a really knowledgeable presentation. I love how thoroughly you've been able to answer our follow-up questions and how engaging you've been with us. And I just really appreciate both of your time. So thank you.
So thank you, Jess, for letting me put you on the spot like that. Awesome. Well, let's give more of our like applause and thanks again, gratitude to our guests, Jade and Mack, for spending time with us this Saturday morning or Saturday afternoon. And if you put your get back in the chat, definitely I'll save it. But if you don't have time to do that, I'll save it at the end of class. So, but yeah, thank you, and thank you for the guests that came to visit us too. So. So thank you. We wish you like all the best with continuing this work. And I'm sure students will stay in touch. Maybe like internships and stuff like that could be possible. We can figure out like research or projects, things like that. Folks want to do that maybe later on as we get into the winter or spring. And keep learning from them and we'll stay in touch. Thank you all. Thank you all for the work you're doing.
Thank
Yeah,
you.
thanks so much.
Thank you. Have a great day.
Thank you
Yeah,
very
have a
much.
great
Thank
day.
you.
Thank you.
So folks in my class, let me see, okay, I can remove your name now too. We're gonna take a break now, so thank